everyone. Today we will discuss about the spectrophotometer. Okay, so let's begin with the class. So what is spectrophotometer? Spectrophotometer is a technique in which the absorption or transmission property of any kind of material is quantitatively measured as a function of wavelength. Okay, so at a particular wavelength, the substance absorbing the light and transmitting the light, okay, on a particular wavelength, that based on that property we can identify, we can quantitatively measure the material. Okay, so that is called as the spectrophotometry study. Okay, and which instrument perform this experiment, that is called as the spectrophotometer. Okay, so the basic principle behind this method is each compound observe or transmit certain kind of light over a certain range of the wavelength. Okay, so suppose this is the material. Okay, this is the material. Okay, so when light passes from this kind of material, then on a particular wavelength it will observe certain kind of certain amount of the light, and remaining amount of the light it will transmit. Okay, so whatever light it will observe and whatever light it will transmit on a particular wavelength, that determines certain parameter for any particular kind of the this matter. Okay, means if suppose there is a lots of matter is present, okay, material is present, the concentration is high, then what will happen is the absorbance of light will be also very high. Okay, and transmission of light will be very less, because there is more substances which observe maximum light and allow the transmission of very small light. Okay, so from that you can, what you can identify is that the what is the concentration present in the solution. Okay, so each compound observe and transmit certain light over a certain range of the wavelength. This principle is performed by the spectrophotometer instrument. Okay, so based on the wavelength of light used, it can be classified into either visible spectrophotometer, UV spectrophotometer, or IR spectrophotometer. Okay, there are three kind of spectrophotometer based on their light absorption property on a particular wavelength. Okay, so visible spectrophotometer, what it does is it absorb the light, or it uses the visible range of the light. Okay, that is the 400 to 700 nanometer of electromagnetic radiation spectrum. Okay, that range you have to remember, that is the 400 to 700 nanometer electromagnetic radiation it will absorb the light. Okay, and this visible spectrophotometer vary in accuracy. Okay, yeah, so whatever light it will absorb, it will vary accordingly. Okay, then for that purpose you can either use the plastic and glass cuvette as well. Okay, for the visible light spectrophotometer. Okay, so this is the visible spectrophotometer. In this you uses the instrument uses 400 to 700 nanometer electromagnetic radiation spectrum. Okay, and for that purpose we can use the plastic as well as the glass cuvette. Okay, so in the the colorimetry is one of the example of visible spectrophotometer. But in the colorimetry, what is happening is it is only color compound. It only means measure the quantitatively measure the colored compound of substances. Okay, but in case of visible spectrophotometer you can means measure the normal kind of substances which is transparent itself, that also you can measure. Okay, so that is the difference. Means colorimetry is very narrow type of instrument, comes under the visible spectrophotometer. In visible spectrophotometer you can also use the transparent material. Okay, then you are having the UV spectrophotometer. So it uses light over the UV range, as name suggests. It uses the light of UV range, that is the 180 to 400 nanometer. Okay, and this UV spectrophotometer is used for the fluid and even solid material also we can use, and only made up of quartz are used for the placing the sample. Okay, so this UV spectrophotometer, it generally uses the UV range light 180 to 400 nanometer. Okay, and like unlike visible spectrophotometer, here the quartz cuvette only can be used. Okay, whereas here the plastic and glass cuvette can be used. Here the quartz cuvette is required. Okay, for UV light determination. Okay, and the third one is the IR spectrophotometer. Okay, so IR is nothing but it is the infrared spectrophotometer, which uses the light of infrared range, that is the 700 to 15,000 of electromagnetic radiation spectra. Okay, so that is all about the three different type of basic type of spectrophotometer. Okay, and here we will see the in the more detail. Okay, so the infrared spectrophotometer often uses, as we have seen, it is the used in the transfer infrared spectroscopy. Okay, it is a technique that measure the absorption of infrared light, okay, by a sample. Okay, so it absorbs the infrared light, okay, that is the 700 to 15,000 nanometer range. Okay, and it is valuable tool for the studying the chemical composition and the its functional group within the protein or any kind of carbohydrate, any kind of lipids. Okay, so what it will do is it is means helpful for identifying the chemical composition of any kind of compound as well as its functional group. Okay, means it is helpful in the functional group identification mainly. Okay, so it can provide information about the type of bond and chemical groups present in the protein structure, it can identify. Okay, not only protein but it can also identify in carbohydrate and lipid, the functional group it can identify. Okay, then second one is the fluorescence spectrophotometer. The fluorescence spectrophotometer is used to measure the fluorescence emission from a sample after it has been excited by a light of a specific wavelength. Okay, it can use to study the presence of fluorescent molecule or fluorophore within a protein. Okay, so this fluorescence spectrophotometer is uses the fluorescence emission. Okay, from a sample. So suppose in a protein, in case of protein, if we will see, the fluorophore molecule are the internal fluorophore molecule are the aromatic amino acid, the phenylalanine, tyrosine and tryptophan. Okay, so these particular amino acid gives fluorescence on a particular wavelength like 280 nanometer. Tryptophan is shows maximum emission. Okay, so that type of fluorescence means by using this type of fluorophore we can identify the presence of this fluorescent molecule inside the protein. Okay, like that. And if you wanted to measure, if you in your protein there is no internal fluorophore, then you can means using the different artificial fluorophore you can means find out the different properties of the conformational changes of protein. Okay, because if your protein's conformation is changed, then your fluorophore molecules location will also change. Like if it is present on the surface of the protein, then it will give a higher intensity wavelength. If it is present in the buried region of the protein, then it will give the low fluorescence intensity. Okay, so on that basis you can find out the location of fluorophore molecule. Okay, as well as the conformational changes inside the molecule. Okay, then you are having the circular dichromism spectrophotometer. So a circular dichromism spectrophotometer is specifically designed for studying the chiral properties of molecule. Okay, including their secondary and tertiary structure. Okay, so it measures the differential observation of light and right circularly polarized light by optically active molecule. It can provide information about the amount of alpha helix and beta city structure present in the protein, which is crucial for the understanding of its secondary structure. So circular dichromism in spectrophotometer, what it does is it is helpful to identify the secondary structure of any kind of protein. Okay, it is basically means most of the time we generally used this instrument for the identification of secondary structural content present in the protein. Okay, and like it is based on the chiral properties of any kind of molecule. Okay, like you know that the amino acid, all the amino acid are chiral in nature. Okay, except the glycine. Okay, so but in case of carbohydrate and lipid, what is happening is there is the chirality is very less comparative to protein. That's why we generally not use any kind of carbohydrate related property and the lipid related property through the circular dichromism spectrophotometer. Only protein we will identify either secondary structure and either secondary structure or either tertiary structure you can find out, but mainly we find out the secondary structure like the alpha helix, the beta structure of the protein we generally find out through circular dichromism spectrophotometer. Then you are having the UV visible spectrophotometer. Okay, so in this UV visible spectrophotometer measure the absorption of light in the ultraviolet and as well as the visible region of the electromagnetic spectrum. Okay, so it is made up of two different type of spectrophotometer range. Okay, so firstly it can identify UV as well as the visible range of the light. Okay, and it is used for the quantitative analysis of the concentration of substance. Okay, and in case of this UV visible spectrophotometer, it is not limited based on the color based measurement. It can access the absorption of light by a wide range of compound, including those that do not produce a distinct color change. Okay, so yeah, here like in case of visible spectrophotometer, um, um, like colorimetry comes. Okay, here also the colorimetry comes, but it can also not limit to the color based measurement. Okay, it can also identify wide range of compound, including those which is transparent in color, which is not produce any distinct color. That also the concentration we can identify through the UV visible spectrophotometer. Okay, so yeah. So here one question it has asked, like the helix content of a protein can be determined using an infrared spectrophotometer, a fluorescence spectrophotometer, a circular dichromism spectrophotometer, or a UV visible spectrophotometer. So here it is asking about the helix content, and a helix is a kind of secondary structural content. Okay, so in the previous slide we have seen that circular dichromism spectrophotometer is generally used for the secondary structural identification of protein. Okay, so this will be the correct answer. Okay, whereas this infrared spectrophotometer is generally used for the functional group identification and the chemical bonding identification. Okay, whereas this fluorescence spectrophotometer we generally used for the location of fluorophore molecule inside the protein or inside any kind of molecule. Okay, but basically in protein molecule. Okay, so this is about the fluorescence spectrophotometers. It is useful for the conformational changes and the location identification of fluorophore molecule. Okay, then you are having a UV visible spectrophotometer, so it uses the UV range as well as the visible range light by the sample. Okay, absorption, and it can useful for the concentration measurement of any kind of substance. Either it can be color based substances or as well as the transparent substances. Okay, so helix content we generally identify by using a circular dichromism. Okay, so that will be the correct answer. Okay, so yeah.